Good morning, good afternoon, everyone. Welcome to our webinar on lowering lead limits on faucets and other endpoint devices, reducing the Q criteria in NSF ANSI CAN 61. We'd like to thank you all for joining us today. My name is Elvan Yalçın. I'm the Marketing Manager EMEA for the Water Division at NSF International, and I'll be doing a quick introduction, and then we'll leave the floor to our expert, Catherine Foster. Catherine Foster is the Technical Operations Manager for Water Distribution Systems at NSF International. Catherine has over 10 years of experience overseeing the review and evaluation of products to NSF ANSI standards 61, 372, and 50, and is the current voting member for NSF International on the Joint Committee on Drinking Water Additives. Catherine holds a Bachelor of Science degree in Microbiology and Molecular Genetics from Michigan State University, and a Master of Science in Cell and Development Biology from the University of Michigan. And today she will be giving an update on recent changes made to the lead extraction requirements for faucets and other endpoint devices under the NSF ANSI CAN 61 standard. You are all muted, but please feel free to type in your questions during the presentation in the chat box. You can select the icon, the arrow on the screen, to display the options. Then you can type your question and click on send button. We'll have the Q&A session at the end of the webinar to answer all your questions. Now I give the floor to Catherine to walk you through the agenda and start the presentation. Floor is yours, Catherine.
Thank you, Elvan. Good afternoon, good morning, good evening, depending on where you're joining us from. I would like to talk today about a reduction in this Q statistic that is used to evaluate faucets for lead extraction. So under NSF 61, faucets and other endpoint devices are evaluated using a calculation known as a Q statistic. After giving background on NSF and NSF 61 generally, I'll describe this Q calculation for lead extraction from mechanical plumbing devices in section 9. And then I'll go on to describe the reduction in the Q criterion from its current 5 micrograms to its new 1 microgram criteria, and show you how this change will be implemented under the standard. And then I'll end with talking about the next steps required for section 9 clients to have their products evaluated to this new criteria. And then we'll have time at the end for some questions and answers. So just a brief background on NSF International for those of you that may not be familiar. NSF is an independent, not-for-profit organization dedicated to providing services to improve public health and safety. We provide a number of services to our clients. Most relevant for today's discussion are that we oversee and facilitate the writing of standards for public health and safety. And we also test and certify products to those standards, which often includes an auditing component. We also provide consulting and training services to our clients as well, although these are independent services from product certification. NSF has developed over 85 currently active national consensus standards, as well as 95 published protocols. And NSF has ties with a number of both national and international government agencies. We are a Pan-American health organization, World Health Organization collaborating center on food safety, water quality, and indoor environment. And we provide services to thousands of organizations in 180 countries. So just a note about how the NSF standards process works. So NSF serves as an administrator for a number of standards for public health and safety, as I mentioned. That means that we facilitate the consensus bodies that oversee and write the standards. All NSF standards are approved by the American National Standards Institute, ANSI, and a number of standards, including Standard 61, are also accredited by the Standards Council of Canada. These accreditation bodies assure that our standards are being developed in accordance with a consensus process that includes addressing the concerns of all stakeholders, The consensus process requires a public review and comment period for any new standards or any modifications to existing standards. All standard meetings are open to the public, and anyone can submit an issue paper to propose changes to the standard. Standard 61 is overseen by the Joint Committee on Drinking Water Additives. This is a committee that is comprised of 3 equal parts: manufacturers, public health representatives, and users. This committee originally developed the standard in 1988, and they continue to meet regularly to revise and propose modifications to the standard to keep it up to date with the current state of the industry and the current state of science. And the Joint Committee, as I mentioned, functions via equal 3-part voting. So there's equal representation between manufacturers, which would include producers and trade associations, public health officials, including representatives from the US EPA, the Centers for Disease Control, Health Canada, academia, and state and local governments, as well as users, which could be anything from consumers to government officials to specifiers, certifiers, and testing labs like NSF, as well as retailers and non-governmental organizations. So each of these different user groups has equal representation on the Joint Committee to ensure that no one group has an overly large influence on the standard. So this standard committee oversees Standard 61, and they must approve all changes proposed for the standard. In addition to the Joint Committee on Drinking Water Additives, any change to NSF 61 that is approved by the Joint Committee requires further approval by a second committee, which is the Council of Public Health Consultants. This is a committee comprised of 50 regulators and user reps. These are all public health officials and representatives from academia. This group oversees all NSF standards, and they're looking to approve the standards to make sure that any changes to the standards are sufficiently protective of public health. This will become relevant later because these are the committees that have worked on and approved the changes to Standard 61, Section 9 that we'll be talking about today. So getting a little more detailed, I'd like to start talking about NSF ANSI CAN Standard 61 in a little more detail at this point. Specifically today, we're going to be speaking of how faucets and other endpoint devices are tested. So Standard 61 is the American national standard and now the Canadian national standard as well for evaluating the health effects of products used in drinking water applications. The scope of the standard covers all products that could have drinking water contact from source to tap. So anything from an intake screen to a well casing from the source water is covered under Standard 61, all the way up to your faucet and the things right at the tap. Standard 61, although today we're going to be talking just about lead, Standard 61 is concerned with any potential contaminants that may leach from a product, not just lead. So that includes other metals, as well as any organic contaminant that might leach from a product. Standard 61, however, does not address product performance. It's purely a health effects standard. Now, Standard 61 is broken up into a number of different sections that categorize products based on the type of product. So in looking at this list of sections, you can get an idea of the very broad scope of Standard 61. Again, it covers everything from pipes and fittings to coatings, tanks, and liners, meters, valves, filters, process media used in a water treatment facility, as well as joining and sealing materials. But today, we're going to be specifically focused on these products that fall under Section 9 of the standard, known as mechanical plumbing devices or sometimes endpoint devices. These devices specifically are those devices that fall within the last liter of the drinking water system. And specifically today I'll be focusing on the evaluation of lead leaching from these Section 9 products. That evaluation under Standard 61 is done using a statistical calculation known as the Q statistic. In statistical terms, the Q statistic is an exact 90% upper confidence bound on the 75th percentile faucet dosage. So in other words, the Q statistic represents a 90% confidence level that 75% of all faucets from a given faucet line would leach lead at dosages at or below the criterion. The calculation is based on testing a given faucet in at least triplicate. So whenever we do a faucet for a Q calculation, we have to test at least 3 individual faucets, although the manufacturer can choose to test more faucets if they desire. Products are then tested according to a 19-day exposure protocol, which I will show you in a few slides. And over that 19-day period, samples are collected on 9 of those 19 days following a 16-hour dwell with the water. So if you take 9 samples per replicate and multiply that by a minimum of 3 replicates, this means that for every Q calculation, there are a minimum of 27 lead extraction data points that feed into the calculation. When we are ready to calculate the Q, the results are first normalized for the at the tap levels for cold end use that would be expected from that faucet. And then the Q statistic itself is a calculation that combines those 27 or more data points into a single value, which is known as the Q statistic. This Q statistic accounts for a variation in lead extraction over that 19-day period, as well as variability between each of the faucet replicates. And it also includes a correction factor that accounts for the relatively small sample size being tested. And I'll go a little bit more into detail on what this Q statistic calculation looks like in a few slides. So when products are tested at NSF, and specifically when they're tested for lead leaching, they are tested with a formulated test water. So this water is buffered and it is set to certain standardized water quality characteristics. In this Section 9 test in particular, the testing is performed using a pH 8 test water. It has an alkalinity of 500 milligrams per liter, DIC level of 122 milligrams per liter, and it has 2 milligrams per liter of free chlorine added as well. Then the faucet testing, again, it's performed over a 19-day period, and the faucets are simply filled with water changes on a set schedule. And then on 9 of the 19 days, water is collected from these faucets and sent for analysis. The analysis is done using ICP-MS for lead, and then those results we get from the test water are what feed into that Q calculation. So this diagram or graphic shows an outline of how the Section 9 exposure protocol is structured over those 19 days. And it's a little bit strange to look at at first, so I'm going to walk you through it with a little marked up diagram here. So typically these faucets are placed on exposure on a Friday. They get a brief washing rinse period, and then they're filled with water and allowed to sit filled with water for a 72-hour period over the weekend. Then on Monday morning, our lab staff comes in and the water is changed every 2 hours in that faucet, and then filled overnight and allowed to dwell for 16 hours. The same procedure is repeated on the second day of exposure, 2-hour water changes followed by a 16-hour dwell. And then beginning on day 3, the first water for analysis is collected. So on the morning of day 3, the water from this 16-hour dwell is collected, and that is our first lead analysis. Then water is continued to be added and replaced from these faucets on a 2-hour basis with a 16-hour dwell overnight. and then water is collected again the next morning. This procedure repeats for day 5. And then we have another 72, or it's actually a 64-hour dwell over the weekend. And then the pattern basically repeats with water collected for analysis again on day 10, 11, and 12. And then again on 17, 18, and 19. So each of these different flasks here represents an individual lead analysis for one faucet replicate. So this gives us 9 data points. And if you'll recall, there are at least 3 faucet replicates that need to be tested. So this gives us a minimum of 27 data points that feed into the Q calculation. This table here illustrates what some of those data may look like. So we have data on each faucet replicate for each collection day of that test protocol. So these are our 27 data points. Typically, you will see reducing levels of lead extraction over the 19-day period. It may look something like this. So these data are then used to calculate the Q statistic. The Q statistic accounts for, first, the geometric mean of the lead dose from each replicate. So the geometric mean from each replicate is taken. And then the standard deviation between each replicate is calculated in order to account for the variability between faucet replicates. So the greater the variability between replicates, the greater the Q statistic will be for this given faucet family. And then the Q statistic also includes an additional factor known as the K factor that is basically a penalty factor that accounts for uncertainty due to the limited sample size. So the fewer the faucet replicates tested, the greater the Q value will be. So this equation is the actual equation for the Q statistic, and you can just break it down into the 3 main parts that I mentioned. So this portion of the Q factor is the geometric mean of the 3-unit lead doses. And then this factor accounts for both the standard deviation of the between the 3 units, as well as a factor that accounts for that uncertainty due to limited sample size. It's important to note that this second factor can never be less than 1. So no matter how many replicates are tested, you can never reduce the Q calculation below the level of the geometric mean of the 3-unit lead doses. That's an important distinction to make. And then just briefly on the R statistic, it's used pretty infrequently, but you may hear references to the R statistic, or sometimes known as the retest statistic. This statistic can be used when a faucet line has failed the Q statistic. So the standard gives this provision that, let's say your first 3 faucets fail the Q statistic. The standard gives a provision for at least 3 additional faucets to then be tested and have those results combined with the results from the initial test, giving a minimum sample size of at least 6. Those accumulated faucet dosages are then used to calculate the retest statistic R. So R is calculated in a similar manner to the Q statistic, except for that the R is an exact 99% confidence bound on the 75th percentile, or 99% confidence that 75% of all faucets from a given faucet line would leach lead at dosages at or below the criterion. And the criteria for the R statistic are the same as the Q, which are currently 5 micrograms and 3 micrograms. So in summary, the overall Q statistic is a representation of the dose or the mass of lead that is imparted to drinking water from a faucet or another mechanical plumbing device. The current criteria for the Q are shown here. So faucets currently are allowed to extract a Q criterion of up to 5 micrograms for faucets and 3 micrograms for flex connectors, stop valves, or other miscellaneous components in that last liter of the drinking water system. The standard has recently been updated to reduce this criteria. So faucets will be held to a 1 microgram mass or dose of lead, and flex connectors, stop valves, and components will be held to a 0.5 microgram Q statistic for lead. So I'm going to talk a little bit now about this change to the standard and the background for its the impetus for the change. So first, there is a clear need for a change. As you all probably know, there is no safe level of lead in drinking water, and reducing lead levels in drinking water has been an evolving process over many past decades. There have been sort of ongoing and continual pushes to reduce the amount of lead that is present in our drinking water. In 2016, the American Academy of Pediatrics issued a recommendation that state and local governments in the US take steps to ensure that water supplies in schools and daycares do not exceed concentrations of 1 microgram per liter of lead. And schools and consultants in the US at least have been working to remediate lead levels under a 3Ts program, and they've been looking for ways to identify lower lead emitting faucets, because even those certified to standard 61 could potentially be leaching a dose of lead up to 5 micrograms into the drinking water. So in 2018 and 2019, task groups or smaller working groups under that NSF ANSI CAN 61 Joint Committee works to develop additional, more stringent criteria for lead release from endpoint devices. Additionally, in February of last year, a bill was introduced to the California State Assembly that would require that only faucets that meet a Q statistic of 1 microgram or less would be able to be sold into the state. So this really galvanized the effort to decide on a change to NSF ANSI CAN 61 and to move to quickly update the standard. So last spring, the Joint Committee and the Council of Public Health Consultants approved changes to the Q criterion in NSF 61. These changes were subsequently published in the 2020 version of NSF 61 in August of 2020. The updates to the standard occur across 2 different sections of the standard. The first one is shown here, and this section lists a series of optional lower lead requirements that manufacturers can choose to have their products evaluated to immediately. The update gives manufacturers the option, for now, to have their product evaluated and test the lower Q criterion of 1 microgram or 5.5 micrograms immediately. Additionally, products that are evaluated to the new criterion must indicate that they comply with the new criterion on their consumer-facing product packaging. This is in order to distinguish newer compliant stock from older stock with the same trade name that may still be available on store shelves. The second update appears in a separate annex section of the standard, and it shows that in 3 years, additional changes to the standard will be made, which will make the optional criteria for low lead become mandatory for all Section 9 devices. So these are outlined in informative Annex 7 of the standard, and specifically, there's a note in this section saying that the optional requirements for lead leaching shall be removed, and the revisions of this annex shall be incorporated into the body of the standard on January 1st of 2024. This date is based on the date of product manufacture. So what we have now is a series of optional requirements where manufacturers can elect to be certified to the lower lead criteria now. But on January 1st of 2024, these criteria will become mandatory, and all products must meet those lower Q criterion in order to remain certified to standard 61. So just to summarize, The 2020 version of Standard 61, which was just published, now includes an optional certification to a reduced Q statistic of 1 microgram for faucets and 0.5 micrograms for flexible plumbing connectors, supply stops, and miscellaneous components. Products that are listed to this new criteria must bear a note both in NSF's product listing, and also the compliance with the new criteria must be noted on consumer-facing product packaging or labeling. So this might be the box that the faucet comes in or a product insert that comes with the faucet. But there must be some way for consumers to differentiate compliant product from older stock that may not comply. In addition to the NSF mark, this product packaging must indicate the following text. It's going to indicate that either the Q is less than 1 or the Q is less than 0.5, depending on whether it's a faucet or one of those other stop valves or component categories. Then on January 1st of 2024, these optional requirements become mandatory for certification to NSF 61, and all faucets must be demonstrated to meet those criteria in order to stay certified to Standard 61. The product labeling requirements will continue to be required even past that January 1st, 2024 date. So if you are a currently certified NSF client and you make these endpoint devices that are present in the last liter of drinking water, that's certified to Section 9, what should you do next? So again, manufacturers may choose to optionally list their product to the new criteria until December 31st of 2023. But in order to do this, NSF will need to reevaluate all product family relationships in order to determine what products comply. As you may know, products are often tested with a test representative that represents a family of other products. So in order to determine whether that entire family complies with the reduced Q criterion, NSF will need to reevaluate those family relationships in order to determine exactly which models comply with the reduced Q criteria and which may need more testing or more information. As I've said, in some cases, additional testing will be required in order to demonstrate that products do indeed comply with that new criterion. In order to be listed to those new criterion, once found to be eligible by the technical review, the consumer-facing product packaging or labeling will need to be updated for all compliant products. And it's important to note that it's very likely that some products may not currently meet the Q criteria, and in order to do so, the products may require a redesign or a swapping out of materials in order to become compliant to the new criteria. So it's important, even if manufacturers are not looking to have their products listed to these lowered criteria now, it's important to begin thinking about what steps may be needed in order to make those products be compliant before the January 1st, 2024 deadline. Because in some cases, it's not only a re-review and a retest that's required, but the products may actually need to be redesigned in order to meet this criteria. Again, by January 1st of 2024, all Section 9 products must have a completed review and possibly additional test data that demonstrate compliance to the new criteria. Any products that are not compliant or not demonstrated to be compliant by that deadline will lose their NSF 61 certification. If you are a currently certified client and you want to know how to get started with this, please contact your NSF account manager, who can give you detailed options for the next steps that you can take to complete this required review. So that concludes my presentation for today. Following this presentation, of course, we'll have time for questions and answers, but if you have additional questions afterwards, please contact your NSF account manager if you're a current client, or if you're interested in getting certification to NSF 61, please contact Vincent Box at the email address shown. So with that, we have some time for questions and answers. Thank you.
Thank you, Katie, for this great presentation. Now we're moving to the Q&A session. We'll be answering some of the questions now, and for the ones that we won't be able to address here, we'll be following up with you after the webinar. So I start with one of them. Will the same legislation also come for Section 8 and other sections?
Yes, so on the legislation, it's unclear how that legislation in California is going to work out right now. It's still very much going through that governmental process. The original bill was actually dropped, but a new bill has been introduced into the California Assembly. So on that bill specifically, it's unclear how that's going to turn out or what the details will be, which is partly why I didn't discuss it much today, because it's still very much a work in progress. So far, that bill is very specific to these Section 9 endpoint devices. And I would be surprised if the bill were extended to any other sections, just given that the Q calculation is only used for Section 9 products. Section 8 and other sections of the standard use different math for evaluating lead extraction. They don't use that Q statistic.
Okay, thank you. What percentage of current endpoints is above the new threshold?
It's hard to tell exactly because, again, our faucets do not represent we don't have a one-to-one relationship between a test and a faucet. We often have one faucet family that's a worst case test, and it represents lots of other families, and it can be very complex. We did look at our faucet tests over the past 10 years, and about 75% of faucet tests did comply with the new criterion. But that's a little bit different than saying that 75% of all faucets comply with that new criterion, because it doesn't take into account those complicated family relationships. So that's part of the reason why these reviews are required. That said, there are many positive signs that most products can comply with these new criteria without significant additional effort.
Okay, thank you. What is the key component leading to lead leachables? Sorry, is it the faucet valves? This is one of the questions. What can be the key reason leading to lead leaching?
Sorry, Alvin, it was cutting out for a little bit of the last part, but I think you said, what's the key components of faucets leading to lead leaching?
Exactly. Can it be the faucet valves? Is the question.
It could be the faucet valves. You know, it really depends on the model of faucets and the way that they're designed. Brass is the only component of faucets that are allowed to contain lead. So typically we see more lead leaching with the greatest surface area to volume ratios of lead in the faucet or of brass in the faucet. I'm sorry, I can't look at myself and answer a question. Yes, so faucets that have a higher surface area to volume ratio of brass or higher lead content brasses typically will tend to extract greater levels of lead. Also, machining of those high lead surfaces, any machined surfaces, tends to cause smearing of the lead on the surface, which can cause greater lead extraction from those faucets.
Okay, great, thank you. Will the same, no, I actually asked that, sorry. In your opinion, will these new requirements open possibilities for brass to plastic replacement?
Certainly, I can't speak to the performance or structural integrity of those types of products, because again, that's not typically something addressed by Standard 61. But if those are feasible replacements, that is certainly an excellent way to remove any lead extraction potential from your products, or at least minimize it to the maximum extent possible, for sure.
Okay. California AB 100 2021 legislative bill. would require earlier implementation of Q1? I don't see the rest of the question. Is it a question? Would it require early implementation maybe?
Yeah, so the most recent version of California AB 100 that we've seen would require implementation by January 1st of 2022, but that is a very aggressive timeline because many of these products, again, will need to be reviewed and retested. And so hopefully the later versions of the bill will take some more time for implementation into account. But the 61 implementation date will be January 1st of 2024, regardless of what's in that California bill in the end.
Okay, thank you. And I'll choose one last question. What is the difference between the Q statistic and the NSF ANSI CANS 372 lead content tests?
Great question. So it's important to differentiate. This test with the Q statistic is about lead leaching from faucets, where we are actually testing how much lead leaches from the faucet into drinking water. NSF ANSI CAN 372 is a lead content standard. So that standard is concerned not with lead leaching, but with the actual lead content from the wetted surfaces of a given faucet. So for that standard, we're simply doing a lead digest and determining whether the lead content of the wetted surfaces meets a maximum weighted lead content average of 0.25%. So that's not affected by these Q criteria reductions.
Okay, I still see questions coming. Can I ask one more?
Absolutely.
Okay. Is there a different calculation regarding lead percentage that a manufacturer can do before submitting the faucet to the leach testing?
So the lead content calculation, the 372 calculation that I just mentioned, that can certainly be done by a manufacturer because it's just based on the maximum specified lead content of the wetted materials used in the product. And that calculation is actually outlined in standard 372 in some detail. The Q calculation is more difficult to estimate upfront by the manufacturer because it's purely based on that lead leaching test. So the manufacturer would really have to try to replicate that 19-day protocol lead leaching test in order to approximate what their Q calculation would be. That said, if you are a currently certified manufacturer, your technical review will give you updates on what your most recent Q results were. And we also offer test-only testing services at NSF, where we will test products just for research and development purposes. So we could test a faucet and give you a calculated Q value for that outside of any formal changes to your product. So that's another option as well.
Okay, great. Thank you so much, Katie, for your time and answering the questions. We still have some unanswered questions, but as I mentioned, we'll be following up with the people after the webinar. Thanks for joining us today and contributing to the discussion with your valuable questions. We will be sending you a very short follow-up questionnaire right after the webinar when you close the window. You'll see the questionnaire. Please fill this out, and in order to give your feedback and your ideas on next webinars. You'll be receiving the recording of this webinar also tomorrow by email. Thank you again and have a good day and stay safe, everyone. Bye.
Thank you.
Thank you. Bye.